January 2022. A company launches with $3 billion. The backers include Jeff Bezos, the richest man alive. The mission: reverse aging. Not slow it, reverse it. Turn back the biological clock at the molecular level. This is not science fiction. This is happening now. And the question you need to ask yourself is simple. How long do you need to stay alive to benefit from what is coming? The answer may be closer than you think. The dream that never dies. But before we begin, do not forget to subscribe, and if you truly enjoy these videos, please leave a like. Let us begin. What you're about to discover will fundamentally change how you think about your own mortality. We're going to explore the laboratories where scientists are reversing aging in mice, the boardrooms where billionaires are betting fortunes on living forever, the cryogenic chambers where hundreds of humans float in liquid nitrogen, waiting for a future that might bring them back, the supercomputers being designed to one day hold the contents of a human mind, and the timeline, the actual timeline that researchers believe separates us from a world where death becomes optional. This is not science fiction. This is happening right now. January 2022. A biotechnology company called Altos Labs launches with $3 billion in funding. The backers include Jeff Bezos, the richest man on the planet. The mission statement is simple and staggering. Reverse cellular aging in humans. Not slow it, reverse it. Turn back the biological clock at the molecular level. The same month, another startup called Retro Biosciences secures $180 million from OpenAI founder Sam Altman. Their goal: add 10 healthy years to human lifespan, not through diet and exercise, through direct intervention in the aging process itself. These are not fringe operations run from garages. These are serious scientific endeavors staffed by Nobel laureates and funded by technology titans who have already transformed how we communicate, shop, and understand the world. Now they want to transform death itself. The question is no longer whether humans will attempt to defeat aging. We already are. The question is whether we will succeed, and how soon. To understand where we're going, we must first understand where we have been. The dream of immortality is as old as civilization itself, perhaps older, carved into clay tablets in ancient Mesopotamia. The Epic of Gilgamesh tells of a king who watches his closest friend die and becomes consumed by terror of his own mortality. Gilgamesh travels to the ends of the earth seeking eternal life. He finds a plant that restores youth. A serpent steals it while he sleeps. He returns home to die like every human before him. 4,000 years later, we are still searching. The ancient Egyptians mummified their dead, preserving bodies for an afterlife they believed was real. The Chinese emperors sent expeditions across the sea seeking islands where immortals were said to dwell. The alchemists of medieval Europe spent lifetimes searching for the philosopher's stone, a substance they believed could transmute lead into gold and grant eternal life to whoever possessed it. Every culture has its version of the fountain of youth. Every generation has its seekers. What separates our moment from all those that came before is not the desire, it is the tools. In 1953, James Watson and Francis Crick described the double helix structure of DNA. They had decoded the language of life itself. Every instruction for building a human body, from eye color to susceptibility to disease to the programmed decay we call aging, written in sequences of just 4 chemical letters. In 2003, scientists completed the Human Genome Project, the entire instruction manual for a human being, 3 billion base pairs, mapped and catalogued. What had taken 13 years and $3 billion to accomplish the first time can now be done in hours for under $1,000. In 2012, Jennifer Doudna and Emmanuelle Charpentier published their breakthrough on CRISPR gene editing, a technology borrowed from bacteria that allows scientists to cut, paste, and rewrite DNA with unprecedented precision, like a word processor for the code of life. In 2006, Shinya Yamanaka demonstrated something that seemed impossible. He took adult cells and reprogrammed them back to an embryonic state. Old cells made young again, not through magic, through the controlled expression of just 4 genes. For this discovery, Yamanaka received the Nobel Prize. Now consider what these tools mean in combination. We can read the genetic code. We can edit it with precision. We can reprogram cells to behave as if they were decades younger. The pieces are falling into place for an assault on aging that previous generations could only dream about. But aging is not a simple enemy. It is a hydra with many heads. Think about what happens to your body over time. Your hair turns grey, your skin wrinkles, your joints stiffen, your eyesight dims, your memory falters, your immune system weakens, your bones become brittle, your arteries harden, your cells divide more slowly and accumulate damage. For most of human history, we accepted these changes as inevitable, the natural order of things, the price of being alive. Scientists now understand that aging is not one process but many. In 2013, researchers published a landmark paper identifying 9 hallmarks of aging, 9 distinct biological mechanisms that contribute to the decay of the body over time. First, genomic instability. Your DNA accumulates mutations and damage with every passing year. Like a document photocopied too many times, the instructions become harder to read. Second, telomere attrition. At the end of each chromosome sits a protective cap called a telomere. Every time a cell divides, these caps get shorter. When they become too short, the cell can no longer divide. It becomes senescent or it dies. Third, epigenetic alterations. Your genes do not change as you age, but the patterns controlling which genes are active and which are silent become increasingly disordered. The conductor of the genetic orchestra loses the ability to keep time. Fourth, loss of proteostasis. Your cells must constantly produce proteins and dispose of old or damaged ones. This quality control system breaks down with age, allowing misfolded proteins to accumulate. These protein clumps are implicated in diseases from Alzheimer to Parkinson. Fifth, deregulated nutrient sensing. Your body uses complex signaling pathways to detect and respond to available nutrients. These pathways, including one called mTOR that we will discuss later, become dysregulated with age, contributing to metabolic dysfunction. Sixth, mitochondrial dysfunction. Mitochondria are the power plants of your cells converting food into usable energy. As you age, these tiny organelles become less efficient, producing less energy and more harmful byproducts. Seventh, cellular senescence. Some cells stop dividing but refuse to die. These senescent cells accumulate in tissues, secreting inflammatory signals that damage neighboring cells. They are like retirees who will not leave the office and spend their time making everyone else miserable. Eighth, stem cell exhaustion. Your body maintains populations of stem cells that can replace damaged tissue. With age, these reserves dwindle. Your capacity for repair and regeneration diminishes. Ninth, altered intercellular communication. Your cells constantly send signals to one another, coordinating their activities. Aging disrupts this communication, leading to chronic inflammation and impaired tissue function. 9 hallmarks, 9 targets for intervention. Scientists are now attacking aging on every front. Some are developing drugs called senolytics that specifically kill senescent cells. In mouse studies, these drugs have extended healthy lifespan by as much as 36%. Some are working to lengthen telomeres, potentially resetting the division counter on aging cells. Some are using gene therapy to reprogram the epigenome, restoring youthful patterns of gene expression. Some are engineering mitochondria to function more efficiently, or replacing damaged ones entirely. Some are growing new organs in laboratories, ready to swap out failing parts like components in a machine. And some are thinking even bigger. Why repair the biological body at all when you might transfer consciousness to a different substrate entirely? Upload the mind to a computer, exist as pure information, potentially forever. The futurist Ray Kurzweil has been making predictions about technology for decades. He foresaw the rise of the internet, the smartphone revolution, and the emergence of artificial intelligence long before most people took these possibilities seriously. His track record is remarkable enough that his current predictions deserve attention. Kurzweil believes we are approaching what he calls longevity escape velocity, the point at which medical advances extend lifespan faster than time takes it away. Add 1 year of life expectancy for every year that passes, then 2 years, then 5. Eventually biological age stops increasing, then it reverses. He puts this moment in the early 2030s, less than a decade away. By 2045, Kurzweil predicts the singularity arrives, the merger of human and artificial intelligence, nanobots flowing through our bloodstream, repairing damage at the cellular level, connecting our brains to the cloud, expanding our intelligence a millionfold. These predictions sound like science fiction, but so did smartphones in 1990. So did mapping the human genome in 1970. Technology advances exponentially, not linearly. Our intuitions, evolved for a world that changed slowly, consistently underestimate what becomes possible. Whether Kurzweil is right about the timeline remains to be seen, but the direction is clear. Humanity is mounting the most serious assault on death in history. The question is not whether we will try to defeat aging. We are already trying. The question is when we will succeed, and what happens to human civilization when we do. The implications are staggering. What happens to retirement when people might live for centuries? What happens to marriage when till death do us part stretches across geological time? What happens to politics when the same leaders could hold power for hundreds of years? What happens to the environment when the human population stops cycling through generations and simply accumulates? What happens to religion when death is no longer inevitable? What happens to meaning when existence stretches toward infinity? These are not hypothetical questions for some distant future. These are questions we may need to answer within our lifetimes, perhaps within the next few decades. In the chapters ahead, we will explore the science in detail. We will meet the researchers extending lifespan in laboratory animals. We will examine the drugs already in clinical trials. We will visit the companies working to reprogram human cells. We will investigate the cryonics facilities where hundreds of people wait in frozen suspension. We will probe the theoretical foundations of mind uploading. And we will confront the ethical and philosophical questions that arise when mortality becomes optional. This is the story of humanity's oldest dream encountering 21st century technology, the dream that never dies meeting the science that might finally make it real. The question you must ask yourself as we begin this journey is simple. If they succeed, do you want to live forever? And perhaps more immediately, how long do you have to stay alive to benefit from what is coming? The answer to that second question may be closer than you think. Let us find out. The biology of aging. The human body is a machine of breathtaking complexity. 37 trillion cells working in concert, each one a microscopic factory performing thousands of chemical reactions every second. Yet this magnificent system has a flaw built into its very design. It breaks down, slowly, inevitably, relentlessly. Understanding why we age is the first step towards stopping it. For decades, scientists argued about whether aging was programmed or accidental. Was it written into our genes as a kind of scheduled obsolescence? Or was it simply the accumulation of random damage over time, like a car rusting in a garage, not designed to fail, but failing nonetheless? The truth turns out to be more complicated than either camp imagined, and more hopeful. Let us start at the beginning. At the level of DNA itself, your genome contains roughly 20,000 genes, instructions for building every protein your body needs. But your genome is not static. It is under constant assault. Ultraviolet radiation from sunlight, reactive oxygen species generated by your own metabolism, environmental toxins, errors in DNA replication. By some estimates, each cell in your body sustains between 10,000 and 100,000 DNA lesions every single day. Your cells possess elaborate repair machinery, proteins that patrol the genome looking for damage, enzymes that snip out defective sections and rebuild them correctly. This system is remarkably efficient. Most damage gets fixed, but not all of it. Some errors slip through, mutations accumulate, the instruction manual develops typos. Most of these changes are harmless. Your body has redundancy built in, but over decades the damage adds up. Critical genes malfunction, cells begin to behave abnormally, cancer risk increases, tissues lose their youthful function. This is genomic instability, hallmark number 1. Now consider what happens at the ends of your chromosomes. Every time a cell divides, it must copy its DNA. But the copying machinery has a limitation. It cannot replicate the very last portion of each chromosome. Without protection, essential genetic information would be lost with every division. Evolution solved this problem with telomeres, long repeating sequences at the chromosome tips that contain no vital information. They exist to be sacrificed. Each division shortens the telomeres slightly. When they become too short, the cell receives a signal. Stop dividing. Your time is up. In some ways, telomeres function as a countdown clock, a cellular hourglass measuring how many divisions remain. There is an enzyme called telomerase that can rebuild telomeres. Stem cells and cancer cells produce it in abundance, but most adult cells do not. Their telomeres shorten inexorably. researchers have experimented with activating telomerase in aging mice. The results were remarkable. Tissue regeneration improved, brain function sharpened, fertility returned, the mice grew physically younger. But there is a catch. Lengthened telomeres also increase cancer risk. Cells that can divide forever are precisely what tumours need to grow without limit. Any therapy targeting telomeres must thread this needle carefully. This is telomere attrition, hallmark number 2. But here is where the story becomes truly interesting. Your DNA sequence is the same in every cell of your body. A liver cell and a brain cell contain identical genes. What makes them different is which genes are active and which are silent. This layer of control above the genome is called the epigenome. Think of your DNA as a piano with 20,000 keys. The epigenome determines which keys get played and which stay silent. Different songs in different cells even though the instrument is the same. The primary mechanism involves chemical tags attached to DNA and to the proteins called histones around which DNA is wrapped. These tags can silence genes or activate them. They can be inherited when cells divide. They constitute a kind of cellular memory. When you were a developing embryo, your cells received precise epigenetic instructions. Become heart muscle here, become nerve tissue there. These patterns persist throughout life, maintaining each cell's identity. But the patterns drift with age. The tags migrate. The wrong genes turn on. The right genes turn off. The cellular symphony becomes cacophony. Cells begin to lose their specialized identities. They forget what they are supposed to be. This discovery led to a profound insight. If epigenetic changes drive aging, and if those changes are potentially reversible, then aging itself might be reversible. In 2006, Shinya Yamanaka demonstrated that adult cells could be reprogrammed back to an embryonic state by activating just 4 genes. These Yamanaka factors, as they came to be known, essentially erased the epigenetic marks that define a mature cell and restored the pristine patterns of youth. The cells did not just behave younger. By every measurable metric, they were younger. This was the spark that ignited the current revolution in aging research. If cells can be reprogrammed to youth, perhaps whole organisms can be as well. In 2016, Juan Carlos Ispizua Belmonte and his team at the Salk Institute took the next step. They applied Yamanaka factors to living mice, not continuously, which would have been dangerous. The mice would have developed tumours as their cells lost their identities entirely. Instead, the researchers used intermittent pulses of reprogramming. The results were striking. Mice with a premature aging condition lived 30% longer. Their tissues showed rejuvenated function, markers of aging reversed. Since then, researchers at Harvard, at Altos Labs, and at dozens of other institutions have been refining the technique. Partial reprogramming, enough to reset the epigenetic clock. Not so much that cells lose their identity. In 2020, David Sinclair's laboratory at Harvard demonstrated that partial reprogramming could restore vision in aged mice. The researchers damaged the optic nerves of old mice, then activated 3 of the 4 Yamanaka factors. The nerves regenerated, sight returned, the mice could see again. More recently, in 2023, Sinclair's team achieved similar results in aged monkeys, and in 2025, Altos Labs published results showing that targeted partial reprogramming extended lifespan in normal mice, not mice with artificial aging conditions. Normal mice living normal lives suddenly living longer and healthier. This is epigenetic alteration, and its reversal. Hallmark number 3. Let us now descend to the protein level. Your cells are protein factories. Ribosomes churn out thousands of different proteins every minute. Each protein must fold into a precise 3-dimensional shape to function correctly. A single misfolded protein is like a key that does not fit its lock. Your cells possess quality control systems. Chaperone proteins help new proteins fold correctly. Autophagy, a recycling process, breaks down old and damaged proteins for reuse. The proteasome, a molecular shredder, destroys defective proteins that cannot be salvaged. With age, this quality control degrades. Misfolded proteins accumulate, they clump together, they interfere with normal cellular function. The tangled masses of tau protein in Alzheimer's disease, the Lewy bodies in Parkinson's, the amyloid plaques that clog aging blood vessels. All are manifestations of lost proteostasis. Researchers are attacking this problem from multiple angles. Drugs that enhance autophagy, gene therapies that boost chaperone production, Techniques to break apart existing protein aggregates. This is loss of proteostasis, hallmark number 4. Now consider how your body senses nutrients. When food is abundant, your cells enter a growth and reproduction mode, build new proteins, store energy, divide and multiply. When food is scarce, your cells shift to a maintenance and repair mode, conserve resources, fix damage, hunker down and survive. The switch between these modes involves several signaling pathways. Among the most important is one called mTOR, short for mechanistic target of rapamycin. When nutrients are plentiful, mTOR is active. It promotes growth. When nutrients are scarce, mTOR quiets down. Here is the crucial observation. Across nearly every organism studied, from yeast to worms to flies to mice, reducing mTOR activity extends lifespan, often substantially. Caloric restriction, the practice of eating significantly fewer calories while maintaining nutrition, activates these same pathways. It is the most reliable intervention known to extend lifespan in laboratory animals. A drug called rapamycin inhibits mTOR directly. In multiple studies, rapamycin has extended lifespan in mice by up to 25%. It works even when started late in life, and unlike caloric restriction, you do not have to be perpetually hungry. Rapamycin is already approved for human use as an immune suppressant in organ transplant patients. But using it for longevity requires careful dosing. Too much suppresses the immune system dangerously. Finding the right balance for healthy aging is an active area of research. Metformin, the common diabetes drug, works through similar pathways. It has been associated with reduced mortality in diabetic patients. A major clinical trial called TAME, targeting aging with metformin, is underway to test whether it can slow aging in healthy adults. This is deregulated nutrient sensing. Hallmark number 5. Inside every cell, thousands of mitochondria generate the energy that powers life. They are the descendants of ancient bacteria that took up residence inside our ancestral cells 2 billion years ago. They still carry their own DNA. They still divide independently. Mitochondria convert glucose and oxygen into ATP, the universal energy currency of cells. But this process is imperfect. It generates reactive oxygen species, highly reactive molecules that can damage DNA, proteins, and cell membranes. Young mitochondria are efficient. They produce abundant energy with minimal waste. Old mitochondria are leaky. They generate less energy and more damaging byproducts. The accumulation of dysfunctional mitochondria contributes to the decline in energy and vitality that characterizes aging. Your cells simply cannot produce the power they need. Researchers are exploring several approaches to rejuvenate mitochondria. Drugs that enhance mitochondrial function, gene therapies that correct mitochondrial DNA mutations, techniques to eliminate damaged mitochondria while allowing healthy ones to proliferate. Recent studies have shown that enhancing a protein called KIAX7RP, which helps mitochondria work more efficiently, extended lifespan in mice and improved their metabolism, muscle strength, and tissue health. The discovery suggests that making mitochondria more energy efficient may be a viable path to slowing aging. This is mitochondrial dysfunction. Hallmark number 6. The remaining hallmarks, cellular senescence, stem cell exhaustion, and altered intercellular communication, we will explore in detail in subsequent chapters. Each represents both a challenge and an opportunity. What matters now is the bigger picture. Aging is not one thing, it is many things. A cascade of interrelated failures that feed on each other. DNA damage leads to epigenetic changes. Epigenetic changes impair proteostasis. Impaired proteostasis damages mitochondria. Damaged mitochondria generate more reactive oxygen. The spiral accelerates. But cascades can work in both directions. If you can reverse one hallmark, you may gain leverage on others. Reprogram the epigenome and cells regain their ability to maintain proteins properly. Clear senescent cells and inflammation decreases, allowing stem cells to function again. Enhance mitochondria, and cells have the energy to repair other forms of damage. This is why researchers are increasingly optimistic. They are not looking for a single magic bullet. They are mounting a coordinated assault on multiple fronts, and they are making progress faster than anyone expected. The question is no longer whether we can slow aging. We already can. The question is whether we can reverse it. And the answer to that question may arrive sooner than you think. The billionaire race against death. In September of 2021, a gathering took place at the Milky Way Research Foundation. The host was Yuri Milner, a Russian-born Israeli technology investor whose fortune was built on early bets in Facebook, Twitter, and other Silicon Valley giants. His net worth exceeds $4 billion. The meeting brought together some of the most influential minds in aging research. They discussed a technology called cellular reprogramming, the ability to turn back the biological clock at the molecular level, make old cells young again. By the time the guests departed, the seeds of the most ambitious longevity company in history had been planted. 4 months later, in January 2022, Altos Labs officially launched. The funding was unprecedented. $3 billion, more than any biotechnology startup in history. Among the investors was Jeff Bezos, whose fortune from Amazon makes him one of the wealthiest humans who have ever lived. Altos did not announce itself with bold promises about extending human lifespan. The stated mission was more measured: understand cellular rejuvenation programming, develop therapies to restore cell health, reverse disease, injury, and disability. But everyone in the industry understood what was really being attempted. The company recruited an all-star roster of scientists. Shinya Yamanaka, whose discovery of reprogramming factors earned him a Nobel Prize, joined as an unpaid senior scientific advisor. Juan Carlos Ispizua Belmonte, whose mouse experiments had demonstrated that partial reprogramming could extend lifespan, became a principal investigator. Steve Horvath, creator of the epigenetic clock that can measure biological age with remarkable precision, signed on. The talent acquisition alone represented hundreds of millions of dollars in signing bonuses and guaranteed salaries. Altos established research institutes in the San Francisco Bay Area, San Diego, Cambridge in the United Kingdom, and Japan. The company created what it calls the Altos Institutes of Science, dedicated to fundamental research, and the Altos Institutes of Medicine, focused on translating discoveries into therapies. For 3 years after its launch, Altos operated in near silence, billions of dollars being spent, hundreds of researchers working, almost no public communication about results. Then in 2025, the first peer-reviewed paper emerged. Scientists at Altos had demonstrated that targeted partial reprogramming could extend the lifespan of normal mice. Not mice engineered with premature aging conditions, not mice with specific diseases, normal, healthy mice living longer because their cells had been made biologically younger. The paper was carefully written. It avoided sweeping claims. But the implications were obvious to anyone who read it. If it works in mice, it might work in larger mammals. If it works in larger mammals, it might work in humans. Meanwhile, Altos had reportedly begun early safety testing in humans, not for life extension. That would require decades of clinical trials, but for specific conditions where cellular rejuvenation might offer therapeutic benefit. Altos Labs is not alone in this race. Retro Biosciences launched with $180 million in seed funding from Sam Altman, the CEO of OpenAI. Altman reportedly invested in his personal capacity, viewing longevity as one of the most important problems humanity could solve. Retro's stated goal is simpler and more direct than Altos: add 10 healthy years to human lifespan. The company is pursuing 3 parallel approaches: cellular reprogramming, similar to Altos; autophagy enhancement, boosting the cell's natural recycling systems that decline with age; and plasma-derived therapies, building on controversial but intriguing research suggesting that factors in young blood can rejuvenate old tissues. By late 2025, Retro was raising an additional $1 billion at a $5 billion valuation. The company announced plans to begin human clinical trials in Australia by year-end. Calico Labs, backed by Google's parent company Alphabet, has been operating since 2013. Founded by Google co-founder Larry Page, Calico took a different approach than the newer entrants. Rather than rushing to therapeutic development, Calico invested heavily in fundamental research into the biology of aging. The company maintains one of the world's largest colonies of naked mole rats. These peculiar underground rodents live up to 30 years, roughly 10 times longer than similar-sized mice. They almost never develop cancer. Understanding how they achieve this remarkable longevity has been a major focus of Calico's research. Calico has also established a partnership with pharmaceutical giant AbbVie, pooling resources for drug development. The company has published important research on reprogramming, on senescent cells, and on the genetics of exceptional longevity. Critics have questioned whether Calico has made sufficient progress given its resources and head start. The company has not yet brought any longevity-focused therapeutic to clinical trials. Defenders argue that fundamental research takes time and that Calico is building foundations that will eventually support transformative therapies. Unity Biotechnology focused specifically on senescent cells. The company's approach was to develop senolytic drugs that could selectively eliminate these aged, dysfunctional cells that accumulate in tissues and contribute to chronic inflammation. Unity attracted significant investment, including from both Bezos and Peter Thiel, another technology billionaire who has been vocal about his desire to live indefinitely. The company advanced a lead candidate called UBX0101 into clinical trials for osteoarthritis. In 2020, the trial failed to meet its primary endpoint. Patients receiving the drug did not show significantly better outcomes than those receiving placebo. Unity's stock price collapsed. The failure illustrated a crucial point. Biological aging is not a simple problem. Even interventions that work dramatically in mice often fail to translate to humans. The path from laboratory to clinic is littered with promising ideas that did not pan out. Unity continues to operate, pursuing other senolytic candidates, but the stumble was a reminder that billions of dollars and brilliant scientists do not guarantee success. New Limit launched in December 2021 with $105 million in initial funding from cryptocurrency billionaire Brian Armstrong, co-founder of Coinbase, and venture capitalist Blake Byers. The company is pursuing epigenetic reprogramming with a focus on developing practical therapies. Life Biosciences, co-founded by Harvard researcher David Sinclair, has developed a platform for partial epigenetic reprogramming. The company's lead program targets ophthalmic conditions, aiming to restore vision in patients with age-related eye diseases. This approach allows clinical testing to proceed more quickly than would be possible for systemic life extension therapies. Sinclair himself has become one of the most visible advocates for the idea that aging is a treatable condition. His 2019 book Lifespan became a bestseller. He has predicted that an age-reversing pill could be available by 2035. Behind these headline-grabbing companies, dozens of smaller startups are attacking specific aspects of aging. Turn Biotechnologies is developing messenger RNA-based approaches to deliver reprogramming factors. Youth Biotherapeutics is working on gene therapies that target aging pathways. Cellgevity claims to have already treated patients with epigenetic reprogramming therapies in jurisdictions with more permissive regulatory frameworks. The company says it has data from over 1,000 treatments. Though this data has not been published in peer-reviewed journals, the total investment flowing into longevity research has accelerated dramatically. By some estimates, more than $40 billion has been committed to the field since 2020, more than was spent in all previous decades combined. Why are the world's richest people suddenly so interested in defeating death? The obvious answer is that they have the most to lose. When you have everything, the only thing money cannot buy is more time. Bezos can afford any material possession on Earth. He has traveled to space. He owns one of the world's largest yachts. But his body is aging at the same rate as everyone else's. His biological clock is ticking. The cynical interpretation is that billionaires are pursuing immortality for selfish reasons. They want to enjoy their wealth indefinitely. They want to maintain their power and influence across centuries rather than decades. The more charitable interpretation is that these investors genuinely believe they can help humanity by funding solutions to one of our oldest problems, that their resources can accelerate research that would otherwise proceed too slowly, that extending healthy human lifespan would be one of the greatest achievements in history. Both interpretations may be true simultaneously. What matters for our purposes is the practical effect. Unprecedented resources are flowing into longevity research. The best scientists in the world are being recruited to work on the problem. Technologies that seemed theoretical a decade ago are now in clinical trials. The billionaire race against death is accelerating the timeline for everyone. Consider the alternative. Without private investment, longevity research would depend primarily on government funding and academic grants. These sources are valuable, but limited. The National Institute on Aging budget is roughly $4 billion per year, spread across thousands of research projects. Altos Labs alone has more concentrated firepower than the entire federal aging research establishment. Private investors move faster than governments. They can take bigger risks. They can offer salaries that attract top talent. They can fund audacious experiments that grant committees would consider too speculative. Of course, there are concerns about who will benefit from whatever therapies emerge. If cellular rejuvenation treatments cost millions of dollars, only the wealthy will be able to afford them. Society could stratify into an effectively immortal elite and a mortal underclass. The rich would accumulate not just wealth, but time itself, compounding their advantages across centuries. The history of technology offers some reassurance. Many innovations that were initially available only to the wealthy eventually became accessible to everyone. Smartphones, commercial air travel, advanced medical treatments. The trajectory of genome sequencing is instructive. The first human genome cost $3 billion to sequence. Today it costs a few $100s, a reduction of 7 orders of magnitude in 2 decades. If longevity treatments follow a similar curve, what costs millions today might cost thousands in 20 years and be covered by insurance in 40. But this outcome is not guaranteed, and the political and social implications of radical life extension will need to be navigated carefully regardless of cost. For now, the race continues. Billions of dollars are being spent. Hundreds of researchers are working. Animal experiments are generating increasingly dramatic results. Human clinical trials are beginning. The first generation of people who will benefit from these investments is already alive. They just do not know it yet. Whether they will gain 5 years or 50 years, or functional immortality depends on how fast the research progresses, and on how long they can stay alive to take advantage of it. The billionaires are betting their fortunes on speed. They understand that the difference between being first and being second in this race is the difference between living forever and dying like every human before them. For them, this is not an abstract investment thesis. It is personal, cellular reprogramming and the fountain of youth. In 2006, a Japanese scientist named Shinya Yamanaka did something that biologists had believed impossible. He turned back time at the cellular level. The experiment was elegant in its simplicity. Yamanaka took ordinary adult mouse cells, skin cells called fibroblasts. Nothing special about them. Then he introduced 4 genes, 4 transcription factors that are normally active only in embryonic cells. The results defied decades of biological dogma. The adult cells transformed. They shed their specialized identity. They reverted to a primitive embryonic state. They became pluripotent, meaning they could develop into any cell type in the body: heart, brain, liver, anything. These reprogrammed cells were called induced pluripotent stem cells, iPSCs for short, and they were not just functionally young. By every measurable metric, the epigenetic marks that accumulate with age had been erased. The cells had been returned to their factory settings. For this discovery, Yamanaka shared the 2012 Nobel Prize in Physiology or Medicine, but the full implications of what he had found would take years to unfold. If adult cells could be reprogrammed to an embryonic state, it meant something profound about aging. The decline was not irreversible. The damage was not permanent. The information needed to restore youth was still present in the cell's genome. It just needed to be unlocked. The question became whether reprogramming could be applied to living organisms, not just cells in a dish, whole bodies. This is where things get complicated. Full reprogramming is dangerous. When you transform an adult cell back to an embryonic state, it loses its identity. A skin cell no longer knows it is a skin cell. A liver cell forgets how to do liver cell things. Apply this to a whole organism and you get chaos. Tissues break down, organs fail. Cancer develops as cells begin dividing without restraint. Early attempts at in vivo reprogramming, applying the Yamanaka factors to living mice, confirmed these fears. The mice developed tumours called teratomas, masses of disorganized tissue containing jumbled mixtures of different cell types. The treatment was worse than the disease. The breakthrough came from understanding that reprogramming is not instantaneous. It is a process that unfolds over days. And crucially, the early stages of reprogramming, when cells begin to shed their age-related epigenetic marks, come before the later stages when they lose their identity entirely. What if you could stop the process early? Reset the epigenetic clock without erasing cellular identity? Partial reprogramming. Juan Carlos Ispizua Belmonte and his team at the Salk Institute tested this idea in 2016. They used mice engineered with a premature aging condition called progeria. These mice age rapidly and die young. making them useful models for studying interventions. The researchers activated the Yamanaka factors in these mice, but only intermittently. Short pulses of reprogramming followed by recovery periods. The cells received the rejuvenation signal, but were not pushed all the way back to pluripotency. The results were remarkable. The treated mice lived 30% longer than untreated controls. Their tissues showed signs of rejuvenation. Markers of aging reversed. Importantly, they did not develop tumors. Partial reprogramming worked. Since then, dozens of laboratories have refined the technique. David Sinclair's group at Harvard has been particularly active. In 2020, they published a paper demonstrating that partial reprogramming could restore vision in aged mice. The experiment targeted the optic nerve. In adults, damaged optic nerves cannot regenerate. Injury to these nerves causes permanent blindness. But in embryonic animals, the optic nerve can regrow. Sinclair's team crushed the optic nerves of aged mice, then activated 3 of the 4 Yamanaka factors in the retinal cells. They used a modified version that avoided the one factor most associated with cancer risk. The nerves regenerated. Vision returned. Old mice that had been effectively blind regained the ability to see. The researchers showed that the treatment worked by resetting the epigenetic clock. The retinal cells were rejuvenated to a younger biological state, and this younger state enabled regeneration that was otherwise impossible. In 2023, similar experiments succeeded in monkeys. The higher up the evolutionary tree you go, the more confident researchers become that the approach will eventually work in humans. By 2025, Altos Labs published results showing lifespan extension in normal mice through targeted partial reprogramming. Not mice with artificial aging conditions. Normal mice living their normal lives, extended by cellular rejuvenation. The results were carefully qualified. The extension was modest. The technique required sophisticated genetic engineering. Translation to humans remained uncertain. But the direction was unmistakable. How does partial reprogramming actually work at the molecular level? Remember the epigenome, the layer of chemical tags that sit on top of DNA and control which genes are active. As cells age, these tags become disordered. Genes that should be silent turn on. Genes that should be active fall quiet. The cell loses its coherent identity. The Yamanaka factors are transcription factors, proteins that bind to DNA and influence gene expression. When these factors are activated, they initiate a cascade of changes that progressively erases epigenetic marks and restores the patterns characteristic of young cells. The key insight is that this process can be interrupted. Stop the reprogramming early enough, and you get the benefit of epigenetic rejuvenation without the danger of dedifferentiation. The cell becomes younger while remaining the type of cell it was. Researchers are now working on multiple approaches to achieve this safely in humans. Gene therapy offers one path: introduce the reprogramming factors using viral vectors with control systems that allow activation and deactivation on demand. This approach requires careful engineering but offers precise control. Messenger RNA delivery is another option. Instead of permanently inserting genes, deliver temporary instructions that cause cells to produce the reprogramming factors for a limited time. The mRNA degrades naturally, limiting the duration of exposure. Small molecule drugs represent the holy grail, chemicals that can activate reprogramming pathways without genetic manipulation. Such drugs would be far easier to manufacture and deliver than gene therapies. They could potentially be taken as pills. Several groups are working to identify small molecules that mimic the effects of the Yamanaka factors. Progress has been made in cell cultures. Translating this to whole organisms is more challenging but not impossible. The timeline for human applications remains uncertain. Clinical trials are already underway for specific conditions where reprogramming might offer benefit. Eye diseases are a natural starting point because the eye is accessible. Outcomes can be measured objectively, and partial treatments have been demonstrated in animals. Life Biosciences, David Sinclair's company, has a program called ER100 targeting optic neuropathies. The therapy delivers 3 Yamanaka factors to retinal cells using gene therapy vectors. If successful, it could restore vision in patients with previously untreatable conditions. Broader applications for systemic aging will take longer. The regulatory pathway for a therapy that claims to reverse aging does not really exist. The FDA does not recognize aging itself as a disease. This creates challenges for clinical trial design. How do you measure success in a life extension trial? You would need to follow participants for decades to show that they actually lived longer. No company can sustain that kind of trial duration. No investor would wait that long for a return. The solution may come from biomarkers, measurable indicators that track with biological age and can be assessed in shorter timeframes. Steve Horvath's epigenetic clock, developed in 2013, pioneered this approach. By measuring the pattern of epigenetic marks at specific sites in the genome, the clock can estimate biological age with remarkable accuracy. People whose biological age exceeds their chronological age have higher mortality risk. Those whose biological age is lower live longer. If a treatment can reduce epigenetic age, as measured by such clocks, it provides evidence that biological rejuvenation is occurring. The FDA has not yet accepted epigenetic age as a validated endpoint for clinical trials, but discussions are underway. Other biomarkers are being developed, panels of proteins in the blood that correlate with aging, functional measures like grip strength and walking speed, cognitive assessments, combinations of indicators that together paint a picture of biological status. As these measurement tools improve, clinical trials become more feasible. Instead of waiting to see if participants live longer, researchers can assess whether their biomarkers improve. This accelerates the development cycle dramatically. The most optimistic projections suggest that partial reprogramming therapies for specific conditions could be available within 5 to 10 years, eye diseases first, then potentially other localized applications. Systemic treatments that rejuvenate the entire body are further out. The challenges of delivery, safety, and dosing multiply when you're treating every cell rather than a specific organ. Realistic timelines probably extend into the 2030s or beyond. But consider what even partial success would mean. If we can rejuvenate retinas, we can cure a major cause of age-related blindness. If we can rejuvenate hearts, we can prevent cardiovascular disease. If we can rejuvenate brains, we might halt or reverse Alzheimer's. Each organ system treated is a separate victory against aging. Each victory extends healthy lifespan. Each extension gives people more time to benefit from the next breakthrough. This is the logic of longevity escape velocity. You do not need to cure aging all at once. You need to stay alive long enough for the next incremental advance, then the next, then the next, until eventually the cumulative progress exceeds the rate of decline. Cellular reprogramming is the most dramatic intervention currently in development. But it is not the only one. In the next chapter, we will examine a complementary approach. Instead of rejuvenating aged cells, what if you could simply eliminate them? Clear out the cellular debris that accumulates with age. Make room for fresh tissue. This is the promise of senolytic drugs. They're already in human clinical trials, and some researchers believe they might reach the market even before reprogramming therapies. The race continues. The drugs that kill old cells. Imagine your body as a city, billions of cells working together, building, maintaining, defending. Every day, some cells are damaged beyond repair. In a young city, the waste management system works efficiently. Damaged cells are cleared away, new cells replace them, the infrastructure stays fresh. But as the city ages, something changes. The waste management breaks down. Damaged buildings are not demolished. They linger, and they start causing problems for the neighbors. This is cellular senescence, and it may be one of the most important targets in the fight against aging. When a cell sustains damage that cannot be repaired, it faces a choice. It can die, clearing the way for replacement, or it can enter a state of permanent arrest, still alive but no longer dividing. This is the senescent state. In small numbers and for short periods, senescent cells serve useful functions. They help coordinate wound healing. They prevent damaged cells from becoming cancerous by forcing them to stop dividing. During embryonic development, senescent cells help shape tissues. The problems arise when senescent cells accumulate. As you age, more cells enter senescence, but your body systems for clearing them become less efficient. The senescent population grows, and these cells are not passive. Senescent cells secrete a complex cocktail of inflammatory molecules, growth factors, and enzymes. This secretion is called the senescence-associated secretory phenotype, SASP for short. The term is clinical. The effects are devastating. SASP molecules spread damage to neighboring cells. They promote chronic inflammation. They break down the structural matrix that holds tissues together. They signal surrounding cells to become senescent themselves. The damage propagates outward, like ripples from a stone dropped in water. In aged tissues, senescent cells can comprise 5 to 15% of the total cell population. That might not sound like much, but the inflammatory signals they produce affect far more cells than their numbers suggest. A small population of toxic neighbours can degrade an entire neighbourhood. The association between senescent cell accumulation and age-related disease is now well established. In osteoarthritis, senescent cells and cartilage secrete enzymes that break down joint tissue. In atherosclerosis, senescent cells in artery walls promote inflammation and plaque formation. In pulmonary fibrosis, senescent cells drive the scarring that progressively destroys lung function. In diabetes, senescent fat cells contribute to insulin resistance. In Alzheimer's disease, senescent glial cells in the brain exacerbate inflammation and neurodegeneration. The list extends to virtually every major age-related condition. Senescent cells are not the sole cause of any of these diseases, but they contribute to all of them. This observation led to a provocative hypothesis. What if you could selectively eliminate senescent cells? What if you could clear the accumulated debris without harming healthy tissue? Would the body recover some of its youthful function? The answer, at least in mice, is yes. The first definitive experiment came in 2011 from the laboratory of Jan van Deursen at the Mayo Clinic. Van Deursen's team engineered mice with a kill switch in their senescent cells. When activated by a drug, the switch triggered cell death specifically in cells expressing high levels of a senescence marker. The researchers activated the switch periodically throughout the mice's lives. The results were striking. Mice with cleared senescent cells lived significantly longer than controls. More importantly, they remained healthier longer. They showed less tissue degeneration, less inflammation, better function of organs including heart, kidney, and fat tissue. They were not just living longer, they were living better. The experiment proved the concept. Senescent cell elimination improved health span and lifespan in mammals. But genetic engineering was not a practical therapy for humans. The field needed drugs that could achieve the same effect through pharmaceutical intervention. Senolytics. In 2015, the first senolytic drug combination was identified. James Kirkland at the Mayo Clinic, building on Van Deursen's work, screened compounds that might selectively kill senescent cells while sparing healthy tissue. Senescent cells survive by activating anti-apoptotic pathways that suppress programmed cell death. This is how they linger despite being damaged. Block these survival pathways and senescent cells die, while healthy cells remain unaffected. Kirkland identified 2 existing drugs that together achieved this selectivity: dasatinib, a cancer medication that inhibits certain kinases, and quercetin, a plant-derived flavonoid with multiple biological activities. The combination became known as D plus Q. In mice, intermittent treatment with D plus Q reduced senescent cell burden. It improved physical function in aged animals. It reduced chronic inflammation. Later studies showed it could extend lifespan. The drugs were already approved for human use, though not for aging. This meant clinical trials could proceed relatively quickly. The first human trial of D plus Q was published in 2019. Patients with idiopathic pulmonary fibrosis, a fatal lung disease associated with senescent cell accumulation, received the combination. The trial was small, only 14 patients, but the results were promising. Physical function improved. No serious adverse effects occurred. Since then, more than 30 clinical trials of senolytic drugs have been completed, are underway, or are planned. Trials are investigating senolytics for diabetic kidney disease, for Alzheimer's disease, for osteoarthritis, for frailty in the elderly, for complications of obesity, for recovery from COVID-19 infection. The results have been mixed. Some trials have shown encouraging signals. The Stamina trial, published in early 2025, tested D plus Q in older adults with mild cognitive impairment and slow walking speed, precursors to more serious decline. The trial was small and open label, meaning there was no placebo group. But participants showed improvements in some measures of cognition and mobility. Biomarkers of senescence decreased in some participants, suggesting the drugs were hitting their target. Other trials have disappointed. Unity Biotechnology's UBX0101, a senolytic designed specifically for osteoarthritis, failed its phase 2 trial in 2020. Patients receiving the drug showed no better outcomes than those receiving placebo. The company's stock collapsed. The Failure illustrated important limitations. Senescent cells are heterogeneous. They vary by tissue, by the cause of their senescence, and by individual patient characteristics. A drug that clears senescent cells in fat tissue may not work on senescent cells in cartilage. What succeeds in one disease context may fail in another. The optimal dosing schedule is unclear. How often should senolytics be administered? Too infrequently and senescent cells accumulate between treatments. Too frequently, and you may interfere with beneficial transient senescence. Finding the right rhythm requires extensive experimentation. Patient selection matters enormously. Clinical trials that enroll patients without high senescent cell burden may fail simply because there are not enough target cells to clear. Biomarkers are needed to identify patients most likely to benefit. Researchers are now developing next-generation senolytics designed to overcome these limitations. Fisetin, a natural compound found in strawberries and other plants, has shown senolytic activity with potentially better safety than D plus Q. Several trials are evaluating fisetin for various age-related conditions. Navitoclax, originally developed as a cancer drug, is a potent senolytic but has significant toxicity, particularly to platelets. Modified versions with improved selectivity are in development. CAR T-cell approaches represent an entirely different strategy. Instead of drugs, these therapies reprogram immune cells to recognize and destroy senescent cells specifically. Early animal studies have shown dramatic results. Senescent cell populations reduced by 90% or more. Health benefits lasting months from a single treatment. A team led by Karina Amer at Cold Spring Harbor Laboratory demonstrated that CAR T-cells targeting a surface marker enriched on senescent cells could clear them in both aged mice and non-human primates. The treated animals showed improved metabolic function, reduced inflammation, better tissue architecture. The approach is more complex than small molecule drugs. CAR T therapies are expensive to produce and administer, but they offer the prospect of durable senescent cell clearance with a single treatment. Vaccines against senescent cells are also being explored. Train the immune system to recognize and eliminate them continuously without repeated drug administration. The field is moving rapidly from proof of concept toward practical therapies. When might senolytics reach general availability? For specific indications like idiopathic pulmonary fibrosis or diabetic kidney disease, drugs could potentially be approved within 5 to 10 years if ongoing trials succeed. These are serious conditions where the risk-benefit calculation favors more aggressive intervention. For general aging prevention in healthy adults, the timeline is longer. Regulatory agencies are conservative about approving drugs for people who are not sick. Demonstrating benefit requires large trials over extended periods. The FDA does not currently recognize aging itself as a treatable condition, though there are efforts to change this. The TAME trial, targeting aging with metformin, was designed partly to establish aging as an indication that regulatory agencies would accept. If it succeeds, it could pave the way for senolytics and other longevity therapies to seek approval for the broader goal of extending healthy lifespan. Meanwhile, some individuals are not waiting. Quercetin is available as a dietary supplement. Fisetin can be purchased online. Dasatinib requires a prescription, but can be obtained through various channels. A subculture of longevity enthusiasts is self-experimenting with senolytic protocols, often guided by physicians sympathetic to the cause. This is not medical advice. The long-term effects of senolytic drugs in healthy adults are unknown. Short-term safety in clinical trials does not guarantee safety over years of use. Clearing senescent cells may have unintended consequences that only become apparent with extended observation. But the interest reflects the urgency many feel about aging. Every year that passes without effective intervention is a year lost. Senescent cells continue accumulating. Damage continues mounting. The window for benefiting from future breakthroughs narrows. Whether senolytics will ultimately deliver on their promise remains to be seen. The early results are encouraging but not definitive. Larger trials with longer follow-up are needed. What is clear is that targeting senescent cells represents a fundamentally new approach to medicine, not treating diseases after they arise, addressing a root cause of multiple diseases simultaneously. The downstream effects of senescence touch cardiovascular disease, diabetes, neurodegeneration, frailty, cancer, and more. A single intervention that reduces senescent cell burden could potentially impact all of these conditions. This is the logic of geroscience. attack the biology of aging itself rather than its individual manifestations. Every disease prevented is a victory. Every healthy year gained is an opportunity. Senolytic drugs are one weapon in this arsenal. Cellular reprogramming is another. Together they represent the most advanced fronts in the war against aging. But some researchers are thinking even bigger. What if the biological body, with all its vulnerabilities and decay, is not the only substrate for human existence? What if consciousness could be transferred to something more durable? In the next chapter, we explore the most radical proposal of all: mind uploading, digital immortality, the question of whether you could live forever as information. Mind uploading and digital immortality. What if you could live forever without a body at all? The idea sounds like science fiction. A consciousness transferred to a computer, the self existing as pure information, immune to disease, immune to aging, potentially eternal. This is the concept of mind uploading, whole brain emulation, digital immortality. And while it remains far from reality, serious scientists and engineers are working to make it possible. The theoretical foundation is straightforward. Your mind emerges from your brain. Your brain is a physical system. Physical systems can, in principle, be simulated. Therefore, a sufficiently detailed simulation of your brain would give rise to your mind. Your consciousness would exist within the simulation just as it currently exists within your skull. The practical challenges are staggering. The human brain contains approximately 86 billion neurons. Each neuron connects to thousands of others through junctions called synapses. The total number of synaptic connections exceeds 100 trillion. And neurons are not simple on-off switches. They are complex electrochemical systems with intricate internal dynamics. To upload a mind, you would need to map this entire structure in sufficient detail to capture the information it contains. Then you would need to simulate its operation with sufficient fidelity to reproduce consciousness. Both tasks exceed our current capabilities by orders of magnitude, but progress is being made. The field of connectomics aims to map the complete wiring diagram of nervous systems. In 1986, researchers published the connectomy of Caenorhabditis elegans, a tiny roundworm with just 302 neurons. It took over a decade of painstaking work. In 2024, scientists completed the connectome of an adult fruit fly, approximately 139,000 neurons, nearly 50 million synaptic connections. The project required years of electron microscopy, terabytes of image data, and sophisticated algorithms to trace neural pathways through the resulting images. The fruit fly brain is roughly 1 million times simpler than the human brain. The gap seems insurmountable, but technology advances exponentially. The same imaging and analysis techniques that mapped the fly brain are being scaled up. Researchers have already mapped cubic millimeters of mouse brain containing tens of thousands of neurons. Larger volumes are in progress. A complete human connectome remains distant. Estimates vary widely, but most researchers believe it will require at least several more decades of technological development. Some suggest it may never be fully achievable due to fundamental limits on imaging resolution and computational capacity. Even a complete connectome would not be sufficient for mind uploading. The static wiring diagram tells you how neurons are connected. It does not tell you the strength of each connection, the state of each neuron at any given moment, or the dynamics of how signals propagate through the network. Functional data is needed, information about how the brain actually operates, not just how it is structured. Recording the activity of every neuron simultaneously is currently impossible for large brains. The best existing technologies can monitor perhaps a few thousand neurons at once. To capture the functional state of 86 billion neurons with the temporal resolution needed to track neural dynamics would require recording systems that do not exist. Some researchers propose workarounds. Perhaps you do not need to capture every detail. Perhaps a sufficiently good approximation would preserve the essential features of consciousness and identity. The question is, how much fidelity is enough? Consider an analogy. A photograph captures enough information about a face that you can recognize the person, but it does not capture the three-dimensional structure, the texture of skin, the way expressions change over time. For some purposes, the photograph is enough. For others, it is woefully inadequate. What level of detail is needed to capture a mind? We do not know. We lack a theory of consciousness, precise enough to answer the question. Assuming the technical challenges could be overcome, how would mind uploading actually work? One approach involves destructive scanning. The brain is fixed, sliced into extremely thin sections, and imaged at nanometer resolution. The process destroys the original, but it captures the structural information needed to build a simulation. This approach has actually been used for small brains. The fruit fly connectome was constructed from electron microscope images of thinly sliced brain tissue. Scaling to human brains would require slicing and imaging roughly 1 million times more tissue. Cryogenic preservation might help. Freeze the brain immediately after death. Prevent decay that would destroy information, then slice and scan at leisure. We will discuss cryonics in more detail in the next chapter. A non-destructive approach would be preferable. Scan the living brain without damaging it. This would avoid the philosophical problem of whether the upload is really you or just a copy that thinks it is you. Current brain imaging technologies fall far short of the required resolution. MRI can image structures down to about a millimeter. Individual neurons are roughly 1/100th that size. Synapses are smaller still. Advanced techniques under development might eventually achieve the necessary resolution. High-field MRI, specialized contrast agents, computational methods that extract more information from noisy signals. But non-destructive scanning at synaptic resolution remains speculative. Another approach involves gradual replacement. Instead of scanning the whole brain at once, replace neurons one at a time with artificial equivalents. As each biological neuron is replaced by a functionally identical synthetic neuron, the transition from biological to digital is gradual. There is no single moment of copying. Continuity is preserved. This approach requires technology that does not exist. Artificial neurons that can integrate with biological tissue, nanoscale devices capable of replicating neural function, the ability to interface millions, then billions, of such devices with the living brain. Ray Kurzweil has predicted that nanobots will eventually make this possible. Microscopic robots injected into the bloodstream. They would migrate to the brain, monitor neural activity, and progressively augment or replace biological function. By 2045, he suggests, such technology could enable the merger of human and artificial intelligence. The timeline is highly speculative. Nanotechnology has advanced significantly, but we are nowhere near producing devices sophisticated enough to replicate neurons. The engineering challenges are immense. Assuming mind uploading becomes possible, what would the experience be like? The uploaded mind would exist within a simulated environment. This environment could be designed to resemble the physical world. A virtual body, virtual surroundings, interaction with other uploaded minds or with artificial intelligences, or the environment could be entirely novel. Geometry is impossible in physical space. Direct access to information, the ability to modify your own cognitive processes, experiences beyond anything biological minds can imagine. Time would be flexible. A simulated mind running on sufficiently fast hardware could experience subjective years in objective minutes, or it could slow down, stretching moments into eternities. The relationship between inner experience and outer time would become programmable. Copying would be possible. An uploaded mind is information. Information can be duplicated. You could create multiple versions of yourself, each diverging from that point forward. The philosophical implications are vertiginous. The uploaded mind would not be immune to destruction. Hardware fails. Software contains bugs. Power outages occur. But unlike biological existence, backups would be possible. Your mind state could be saved periodically. If something went wrong, you could be restored from the last backup. Death would become a matter of data loss rather than biological failure. With sufficient redundancy, it could be made arbitrarily unlikely. Would an uploaded mind really be you? This is the deepest question surrounding mind uploading, and there is no consensus answer. If your brain was scanned destructively and a simulation instantiated on a computer, that simulation would believe it was you. It would have your memories, your personality, your sense of continuous identity. From its perspective, it would be you, waking up in a new substrate. But from your perspective, you would be dead. The biological process that was your consciousness would have ended. Something else, something that resembles you, would continue. But the thread of experience that is you would be cut. Some philosophers argue this concern is misguided. Personal identity is not about continuity of substrate, it is about continuity of pattern. If the pattern is preserved, you are preserved, regardless of what instantiates the pattern. Others argue that subjective experience is tied to specific physical processes. A simulation might behave identically to you without actually being conscious. It would be a philosophical zombie, an entity that acts as if it has inner experience but actually has none. These questions cannot be resolved through armchair reasoning. We lack the understanding of consciousness needed to answer them definitively. Perhaps we never will. What is the current state of mind uploading research? The Carbon Copies Foundation is a non-profit organization dedicated to advancing whole brain emulation. They fund research, host workshops, and coordinate efforts across the scientific community. Their work is serious, but their timelines are long. They acknowledge that functional mind uploading is likely decades away at minimum. Academic researchers are making progress on component technologies, better imaging, better simulation, better understanding of neural computation. But integrated systems capable of uploading even simple organisms remain beyond current reach. Commercial interest is limited compared to other longevity approaches. The technical barriers are higher, the timelines are longer, the payoff is more uncertain. Investors prefer bets with clearer paths to return. Nectomy, a startup that emerged from MIT, attracted attention and controversy for its approach. The company proposed to preserve brains using a process called aldehyde-stabilized cryopreservation. The catch was that the process was fatal. It had to be performed while the person was still alive to achieve optimal preservation. The idea was that future technology might be able to extract information from the preserved brain and reconstruct the mind. But since the preservation required death, it was essentially a form of physician-assisted suicide combined with speculative future technology. The ethical concerns were substantial. Nectum paused its service offerings following criticism. The technical feasibility of reconstructing minds from chemically preserved brains remains unproven. For now, mind uploading remains more philosophy than engineering, a thought experiment that illuminates questions about identity, consciousness, and what it means to be human. A possible future, rather than an imminent one. But the research continues. The connectums grow more complete. The simulations grow more sophisticated. The computational power grows exponentially. If minds can be uploaded, perhaps they eventually will be. And a civilization of uploaded minds would be fundamentally different from anything that has existed before. Potentially immortal, potentially unbounded, potentially unrecognizable. Whether that future appeals to you is a personal question, whether it will arrive is a scientific one. The answer is not yet known. But in the meantime, other approaches to immortality are further along. Technologies that preserve the biological body in hopes of future revival. We turn now to cryonics, the science of freezing humans and the bet that death might not be permanent. Frozen in time, Scottsdale, Arizona, a nondescript industrial building in the desert. Inside, rows of massive steel cylinders stand in climate-controlled rooms. Each cylinder is a dewar, a specialized container designed to hold liquid nitrogen at minus 196 degrees Celsius. Inside those dewars, suspended in the cold, are human beings, not alive in any conventional sense, legally dead, but preserved, waiting. This is Alcor Life Extension Foundation, the world's leading cryonics organization, and those cylinders contain over 250 patients who have made the ultimate bet on future technology. Cryonics is based on a simple premise. Death is a process, not an event. When your heart stops, you do not instantly cease to exist. Your cells die gradually. Your brain, deprived of oxygen, begins to deteriorate, but for some period of time, the information that constitutes you remains intact. If that deterioration could be stopped, if the dying process could be paused, then perhaps future medicine could repair whatever damage killed you and restore you to life. Cryonics attempts exactly this. Immediately after legal death is declared, a specialized team begins the preservation process. Blood is replaced with cryoprotectant solutions, chemicals that prevent ice formation, which would shatter cell membranes and destroy tissue structure. The body is cooled rapidly to cryogenic temperatures. At minus 196 degrees, all biological activity ceases. Molecules barely move. Chemical reactions effectively stop. The patient is suspended in time. Alcor calls these individuals patients rather than bodies. The terminology is deliberate. They are not considered dead in the absolute sense. They are in a state of stasis, awaiting treatment that does not yet exist. The concept dates to the 1960s. Robert Ettinger, a physics teacher from Michigan, published The Prospect of Immortality in 1964. He argued that cryogenic preservation was technically feasible with existing technology, and that future science would eventually develop the means to revive preserved individuals. The first person to be cryopreserved was James Bedford, a psychology professor who died of cancer in 1967. His preservation was primitive by modern standards. The techniques have improved dramatically since then. Bedford remains in cryostasis at Alcor, the longest preserved patient. Today, approximately 500 to 650 people worldwide have been cryopreserved. The numbers grow by 30 to 40 annually. Several thousand more have signed up for future preservation. Alcor is the largest and most technologically advanced provider. The Cryonics Institute in Michigan is another major organization, offering preservation at lower cost. Cryorus in Russia, Tomorrow Bio in Europe, and Shandong Yinfeng in China serve additional markets. The cost varies significantly. Alcor charges $200,000 for whole body preservation. The Cryonics Institute charges $28,000. The difference reflects varying approaches to facilities, procedures, and long-term care funding. Most members fund their preservation through life insurance policies. A healthy young person can obtain a $200,000 policy for relatively modest premiums. The policy names Alcor as beneficiary. When the member dies, the insurance pays for preservation. What exactly happens during cryopreservation? The process ideally begins within minutes of legal death. Alcor maintains standby teams that can deploy to members' locations when death appears imminent. The faster preservation begins, the less deterioration occurs. First comes stabilization. The body is packed in ice. Chest compressions and ventilation maintain some circulation and oxygenation. Medications are administered to protect brain tissue. Then comes perfusion. The blood is replaced with cryoprotectant solution. This is a complex mixture of chemicals that penetrates tissues and prevents ice crystal formation. The solution is pumped through the circulatory system, displacing blood and saturating organs. The goal is vitrification rather than freezing. Water in tissues does not form ice crystals. Instead, it transitions to a glass-like solid state. No crystalline structure means no ice damage to cells. Cooling proceeds gradually over several hours. The body passes through dangerous temperature ranges where ice might form if cryoprotection is incomplete. Eventually, the patient reaches liquid nitrogen temperature. At this point, biological time stops. The patient could remain in this state indefinitely, decades, centuries, however long it takes for revival technology to emerge. Alcor stores patients in large dewars that hold multiple individuals. Whole body patients are positioned upside down so that if any liquid nitrogen were to boil off, the head, containing the brain, would remain submerged longest. Some patients choose neuropreservation, where only the head is preserved. This costs less and may be easier to revive if future technology can grow new bodies or transfer minds to other substrates. The fundamental question is whether revival will ever be possible. Critics argue that cryopreservation causes irreversible damage. Even with the best vitrification techniques, ice formation is not completely prevented. Cell membranes are disrupted, proteins denature. The precise molecular arrangements that constitute memory and identity are scrambled beyond recovery. Furthermore, the cause of death must be addressed. A patient who died of cancer still has cancer. A patient who died of heart disease still has damaged cardiovascular system. Revival requires not just undoing cryopreservation damage, but curing whatever killed the person in the first place. Proponents acknowledge these challenges but argue they are engineering problems, not fundamental impossibilities. The information that constitutes identity is encoded in brain structure, synaptic connections, protein distributions, epigenetic marks. As long as this information is preserved, even imperfectly, future technology might be able to read it and reconstruct the original. Consider how medical capability has advanced over the past century. Conditions that were death sentences in 1920 are routinely treatable today. Extrapolate forward another century, another 2 centuries, another millennium. What seems impossible now may become straightforward. Nanotechnology offers one potential pathway: molecular scale machines that could enter preserved tissue, catalogue the damage, and repair it atom by atom. Robert Freitas has written extensively about medical nanorobots and their theoretical capabilities. The technology does not exist yet, but physics does not forbid it. Mind uploading offers another pathway. If consciousness can be transferred to a digital substrate, perhaps the information in a preserved brain could be extracted and instantiated in a simulation. The biological body would not be revived, but the person, in some sense, would continue. Neither pathway is guaranteed. Both require technological advances that may take decades or centuries, or may never arrive. Cryonics is a bet, a wager that the future will be more capable than the present, that humanity will develop the means to reverse what currently appears to be death, that you, specifically, will be among those revived. The odds are unknown. There is no way to calculate the probability that cryonics will work. It depends on too many variables: the progress of technology, the survival of cryonics organizations, the continued availability of resources for long-term storage, the willingness of future societies to revive people from the past. Some estimates place the probability at a few percent, others at fractions of a percent. A small chance, but not zero. Is a small chance of revival worth the cost? For most people, the answer is no. $200,000 could fund many experiences in a known lifetime. The uncertainty is too high, the payoff too speculative. But consider the alternative. Without cryopreservation, the probability of revival is exactly zero. You die, you decay. Whatever information constituted you is lost permanently. Cryonics offers a small positive probability compared to certain zero. From a purely mathematical standpoint, any positive probability times infinite value equals infinite expected value. If you believe extended life has very high value, even tiny odds of achieving it justify the investment. This is Pascal's wager applied to technology rather than theology. Critics point out that the analogy is imperfect. Pascal's wager involves no ongoing cost after the decision. Cryonics requires organizations to survive, dewares to be maintained, and future societies to prioritize revival. Many things can go wrong over centuries of waiting. Cryonics organizations attempt to address these concerns through financial structures. Alcor places a substantial portion of each patient's funding into a patient care trust. This trust invests the funds and uses returns to cover ongoing storage costs. The corpus is intended to grow over time, eventually funding revival research and procedures. Whether these structures will survive the test of time remains to be seen. Cryonics is an experiment without controls. The first generation of patients will not be revived for decades at minimum, perhaps never. In 2025, something remarkable happened. A California court ruled that a cryopreserved individual could be classified as in stasis rather than deceased. The ruling created a third, legal category, neither fully alive nor fully dead. While the implications are still being worked out, the decision acknowledged that cryopreservation is meaningfully different from other forms of death. The scientific mainstream remains skeptical. Most physicians and biologists do not believe cryonics will work. The damage from preservation is too severe. The technological requirements for revival are too speculative. The whole enterprise is viewed as wishful thinking dressed up as science. But skepticism about new technologies has often proved wrong. Heavier-than-air flight was considered impossible by prominent scientists until the Wright brothers proved otherwise. Space travel was science fiction until it was science fact. The human genome was considered too complex to sequence in any reasonable time frame until it was done. Cryonics may be genuinely impossible, or it may be the seed of something revolutionary. For those who choose preservation, the reasoning is often personal. I have nothing certain to lose by trying. I have potentially everything to gain. If it does not work, I will never know. If it does work, I get to continue. The patients in Alcor's dewars cannot speak for themselves. They rest in frozen silence, neither alive nor conventionally dead. Their fates depend on what the rest of us do, whether we develop the technologies they are betting on, whether we choose to revive them if we can. They are emissaries to the future, messages in bottles cast into the ocean of time. Whether anyone will be there to receive them remains the ultimate question. But while cryonics bets on future technology, other approaches aim to enhance the living body now. Technologies that could make biological immortality achievable without ever dying in the first place. We turn next to nanotechnology and artificial intelligence, the machines that might one day live inside us and keep us alive forever. The machines inside us. Ray Kurzweil takes over 200 pills and supplements every day. He has done so for decades. The regimen is designed to optimize his health, slow his aging, and keep him alive long enough to benefit from the technologies he believes are coming. Kurzweil turned 77 in 2025. His chronological age climbs at the usual rate, 1 year per year, but his biological age, he claims, has remained relatively stable. The pills, the careful diet, the measured lifestyle, all buying time. Time for what? Time for the nanobots. Kurzweil has predicted for years that microscopic robots will eventually flow through human bloodstreams. They will monitor health in real time, they will repair damage at the cellular level, they will destroy pathogens, clear arterial plaques, and eliminate cancer cells before tumours can form. These nanobots will connect our brains to the cloud, they will expand our intelligence, they will blur the boundary between biological and artificial. By 2045, Kurzweil believes the merger will be complete. Humans and machines will become one. The singularity. Is any of this plausible? Nanotechnology is real. Scientists have been manipulating matter at the molecular scale for decades. The term was coined by Eric Drexler in the 1980s. His vision of molecular assemblers, machines that could build complex structures atom by atom, has inspired generations of researchers. Progress has been substantial, but slower than early predictions suggested. We can build nanoparticles for drug delivery, tiny capsules that carry medications to specific tissues. Cancer treatments using such particles are already approved and in clinical use. We can create nanoscale sensors, devices that detect specific molecules or conditions at extremely small scales. Some are being developed for continuous health monitoring. We can engineer molecular machines, proteins or synthetic structures that perform mechanical functions, move along tracks, rotate, change shape in response to signals. Biological cells are full of such machines, evolved over billions of years. But the nanobots of Kurzweil's vision, autonomous robots smaller than cells that could navigate the body, perform complex tasks and communicate with external systems, remain beyond current capability. The engineering challenges are immense. Power is one problem. How do you supply energy to billions of microscopic devices distributed throughout the body? Batteries do not scale down well. Harvesting energy from blood flow or body heat is theoretically possible, but practically difficult. Navigation is another challenge. The body is a complex environment. Blood vessels branch into capillaries finer than a human hair. Tissues have varying densities and compositions. Getting nanobots to where they are needed and keeping them there requires sophisticated guidance systems. Communication adds complexity. If nanobots are to coordinate their activities or transmit information, externally they need some form of signaling. Radio waves do not penetrate tissue well. Chemical signaling is slow. Acoustic or optical methods have their own limitations. Biocompatibility is essential. Anything in the body triggers immune responses. Foreign objects are attacked, encapsulated, cleared. Nanobots would need surfaces that evade detection or active mechanisms to suppress immune reactions. Manufacturing at scale is perhaps the greatest hurdle. Even if we could design effective nanobots, producing trillions of them reliably and economically is a manufacturing challenge unlike anything we have faced. None of these problems are impossible to solve. Physics does not forbid nanobots, but solving them will require decades of additional research and development. Kurzweil's 2045 timeline is optimistic by most assessments. More conservative estimates place practical medical nanobots in the latter half of the 21st century. Some researchers believe they may never be feasible in the form Kurzweil imagines, but intermediate technologies are advancing rapidly. Implantable devices are already transforming medicine. Pacemakers regulate heartbeats. Cochlear implants restore hearing. Deep brain stimulators treat Parkinson's disease and depression. Insulin pumps manage diabetes. These are macro-scale devices, not nanobots, but they demonstrate the principle of technological intervention in biological systems. Brain-computer interfaces are progressing remarkably. Neuralink, founded by Elon Musk in 2016, has developed implantable chips that interface directly with brain tissue. The company's device, called the Link, consists of a small processor and flexible electrode threads thinner than human hair. Surgically implanted, it can record neural activity and potentially stimulate neurons. In 2024, Neuralink implanted its device in a human patient for the first time. The patient, paralyzed from the neck down, was able to control a computer cursor using only his thoughts. He played video games. He browsed the internet. The interface translated his neural intentions into digital commands. The initial applications are medical, restoring communication and mobility to people with severe disabilities. But the long-term implications extend further. If we can read neural activity with sufficient resolution, we can understand what the brain is doing. If we can stimulate neurons precisely, we can influence what the brain does. The interface goes both ways. Kurzweil envisions brain-computer interfaces not as assistive devices for the disabled, but as cognitive enhancers for everyone. Connect your brain to artificial intelligence, access information instantly, expand your memory, augment your thinking. This would not require nanobots initially. Current and near-term interfaces use implanted electrodes, cruder than molecular scale devices but already functional. The limiting factor is bandwidth. How many neurons can you interface with simultaneously? Current devices connect to hundreds or thousands of neurons. The brain contains 86 billion. To achieve the kind of deep integration Kurzweil describes would require interfacing with vastly more neurons than current technology allows. Progress is rapid. Electrode counts are increasing. Recording quality is improving. Non-invasive approaches using external sensors are being developed alongside implantable devices. Whether the technology will advance fast enough to meet Kurzweil's timeline remains uncertain. But the direction of travel is clear. Artificial intelligence is another crucial piece. The last few years have seen explosive progress in AI capabilities. Large language models like GPT demonstrated that scaling neural networks and training data produces emergent abilities: reasoning, creativity, explanation. Capabilities that seemed far off arrived suddenly. AI is already transforming drug discovery. Machine learning systems can screen millions of potential compounds for therapeutic activity. They can predict protein structures that would take years to determine experimentally. They can identify patterns in biological data that humans miss. Altos Labs, Retro Biosciences, and other longevity companies are using AI to accelerate their research, analyzing cellular reprogramming, identifying senolytic candidates, modeling the complex interactions that drive aging. If artificial general intelligence arrives, as some researchers believe it will within the next decade or two, the implications for longevity research are profound. An AI system smarter than any human could potentially solve biological problems that have stumped researchers for generations. Design therapies we cannot imagine. Run experiments virtually before testing physically. The synergy between AI and biotechnology may be what ultimately delivers radical life extension. Imagine an AI system that understands biology at a level no human can achieve. It designs nanoscale devices optimized for the human body. It identifies drug combinations that reverse specific aspects of aging. It monitors individual patients continuously and adjusts treatments in real time. It makes discoveries faster than the entire human research enterprise combined. This is the scenario that makes Kurzweil optimistic about his timelines. Artificial intelligence is advancing exponentially. As it becomes more capable, it accelerates everything else, including the development of longevity technologies. The counter-argument is that biological systems are more complex than digital ones. Understanding the body is different from training a language model. Unexpected interactions, emergent effects, and chaotic dynamics may resist even the most powerful AI. Medicine has a history of humbling, confident predictions. Diseases that seemed conquered return in resistant forms. Therapies that work in trials fail in practice. The body is a system evolved over billions of years. It does not yield its secrets easily, but the combination of advancing AI, improving biotechnology, and accumulating biological knowledge is formidable. Each breakthrough enables others. The pace accelerates. Gene therapy is becoming practical. Viral vectors can deliver corrective genes to specific tissues. CRISPR enables precise editing of genetic sequences. Inherited diseases are being cured through genetic intervention. Aging-related genes may eventually be targetable. Organ regeneration is progressing. Stem cells can be coaxed to form specific tissue types. Organoids, miniature organs grown in laboratories, are used to study disease and test drugs. Full-sized organs may eventually be growable from patient cells, eliminating transplant rejection. Senolytics, as we discussed, are in clinical trials. Reprogramming therapies are advancing. Multiple fronts in the war against aging are seeing progress simultaneously. The machines inside us may not arrive as quickly as Kurzweil predicts, but they are coming. Some already exist: implants, pumps, stimulators, interfaces. More sophisticated versions are in development: nanoscale drug delivery, continuous monitoring, brain-computer integration. Artificial intelligence is amplifying human capability, making discoveries faster, identifying solutions that would take humans decades to find. The question is not whether technology will transform human biology, it is when and how much, and whether we can navigate the transformation wisely. The technologies we have discussed offer extraordinary possibilities: extended health, radical longevity, perhaps even immortality. But they also raise profound questions. Questions about fairness, about identity, about meaning, about what it means to be human in a world where the boundaries of humanity are being redrawn. We must address these questions, the ethics of living forever. This is where we turn next. The ethics of living forever. Imagine a world where some people live for centuries while others die at 80, where the wealthy can afford cellular rejuvenation, and the poor cannot, where the gap between rich and poor becomes not just a matter of comfort, but of time itself. This is not science fiction. It is a plausible outcome of the technologies we have been discussing, and it raises ethical questions that humanity has never before faced. Who deserves to live forever? The initial cost of longevity treatments will almost certainly be high. Altos Labs has spent billions on research. Clinical trials cost hundreds of millions. Regulatory approval takes years of expensive testing. When the first cellular reprogramming therapy reaches market, it will not be cheap. Consider the history of other transformative technologies. The first personal computers cost thousands of dollars, accessible only to enthusiasts and corporations. The first mobile phones were luxury items for executives. The first genome sequences cost billions. Over time, costs declined, manufacturing scaled, competition increased. What was once exclusive became ubiquitous. Today, billions of people carry smartphones more powerful than the computers that guided Apollo missions. Longevity technologies may follow a similar trajectory. First available to the wealthy, then to the middle class, eventually universal. A transition measured in decades, not generations. But aging does not wait. During those decades of declining costs, millions of people will die who might have lived. The people born too late to afford early treatments. The people in countries without access to advanced medicine. The people whose lottery ticket in the birth lottery came up short. Is this unjust? In one sense, it is the same dynamic that characterizes all medical progress. New treatments are always expensive before they become cheap. We do not refuse to develop cancer drugs because some people cannot initially afford them. The alternative, halting research until universal access can be guaranteed, would condemn everyone to die of diseases that might have been cured. But longevity is different from treating disease. We have some intuitive acceptance that lifespans vary, that some people live longer than others due to genetics, lifestyle, luck. Extending this variation from decades to centuries changes something fundamental. A society stratified by lifespan would be unlike any that has existed. The wealthy would accumulate not just money, but time. Time to compound their advantages, time to entrench their power, time to shape the world across generations. Imagine a corporate executive who remains in power for 200 years, a political dynasty that spans centuries, an investment fortune that compounds for a millennium. The scale of inequality becomes almost inconceivable. Those on the short end of this divide would face mortality in a way that previous generations did not. Today, death is universal. Everyone faces the same fundamental fate. Rich and poor, powerful and weak, all eventually die. There is a grim equality in this. In a world of differential immortality, that equality vanishes. Death becomes a marker of status, of worthiness, of having been left behind. The psychological and social consequences are difficult to predict. Perhaps those with limited lifespans would rage against the injustice. Perhaps they would accept it as they accept other inequalities. Perhaps religions would evolve that sanctify natural mortality as spiritually superior. Perhaps revolutions would target the immortal elite. The policy choices we make now will shape these outcomes. If longevity technologies are developed primarily by private companies seeking profit, access will follow market logic. Those who can pay will receive treatment. Those who cannot will not. If governments invest in longevity research and ensure public access, The benefits might be distributed more broadly. Universal longevity care, like universal healthcare, could become a social entitlement. International coordination would be needed. A country that offers longevity treatments to its citizens while others do not creates new dimensions of global inequality. Brain drain could accelerate as talented people migrate toward longer lives. The debate over access must begin now, while the technologies are still developing. Once entrenched interests form around profitable exclusivity, changing course becomes much harder. Beyond access, longevity raises questions about identity. Who are you across centuries of life? Personal identity is tied to memory. The person you were 10 years ago shares memories with who you are today. This continuity makes you the same person, but memory is imperfect. You probably cannot recall specific days from a decade ago. You have forgotten most of what you learned in school. The person you were at 5 is connected to who you are now by an attenuated chain of partially preserved experiences. Extend this across centuries. The person you become in 300 years might share almost nothing with who you are today. Different memories, different values, different relationships, different context. Is that really you? Or is it a successor who happens to share some genetic material? The ship of Theseus problem becomes personal. If every plank is replaced, is it still the same ship? If every memory fades and every belief changes, is it still the same person? Perhaps personal identity is less fixed than we imagine. Perhaps continuity is an illusion even in normal lifespans. Perhaps what matters is not preserving a fixed self but enabling continued growth and experience. Or perhaps identity depends on something deeper, a continuous thread of consciousness, a persistent sense of being the same entity across time. If cellular reprogramming or mind uploading disrupts this thread, something important may be lost even as something else continues. These questions have been debated by philosophers for centuries. Radical life extension makes them practical rather than abstract. Then there is the question of meaning. Does life require death to have significance? Many philosophical and religious traditions answer yes. Mortality gives urgency to existence. Because time is limited, choices matter. Because we will die, we strive to create, to connect, to leave something behind. The existentialists argued that confronting death is essential to authentic living. Awareness of finitude forces us to define our own meaning rather than drifting through received assumptions. If death becomes optional, does meaning evaporate? Perhaps not. Perhaps meaning comes from engagement with the world rather than from a ticking clock. Perhaps we can find purpose in growth, creation, and connection, regardless of how long we live. But there is a risk. Infinite time could become infinite procrastination. Why act today if you have forever? Why commit to anything if you can? Always change course later. Why risk anything if you have endless years to lose? The psychology of immortality is unexplored territory. We have no experience with humans who expect to live indefinitely. Depression, ennui, and existential crisis might become more common, not less. Relationships would transform. Marriage until death do us part takes on new meaning when death might never come. Could romantic love sustain across centuries? Would people form shorter-term partnerships with the explicit understanding that forever is too long? Friendships might stratify by age cohort. Those born in the same era sharing cultural references and life experiences that those born centuries apart cannot access. Society fracturing into temporal tribes. Family structures would become unrecognizable. Great-great-great-grandchildren interacting with ancestors who remember the 20th century. Inheritance accumulating across generations. The power of the elderly compounding as their numbers swell. Population presents obvious challenges. If people stop dying and continue reproducing, population grows without limit. Earth's resources are finite. Something would have to give. Possible responses include restricting reproduction, expanding into space, or accepting that longevity treatments will never become truly universal. Each carries its own ethical weight. China's one-child policy offers a cautionary example of reproductive restriction. Coercive population control creates profound human rights concerns. Voluntary restraint might be achievable, but has no historical precedent. Space colonization could theoretically provide unlimited room for an unlimited population. But the timelines are uncertain. Terraforming Mars or building orbital habitats would take centuries even with aggressive development. Population pressure could arrive before escape valves open. The alternative, Accepting that most people will continue to die while some achieve radical longevity leads back to the inequality concerns discussed earlier. Environmental impacts extend beyond population. Longer-lived individuals might consume more resources over their extended lifespans, or they might become better environmental stewards, caring more about a planet they expect to inhabit for centuries. Governance would face unprecedented challenges. Democratic systems assume turnover. New generations bring new perspectives. Old leaders die and new ones rise. The cycle of replacement limits the entrenchment of any particular ideology or interest. If leaders live for centuries, this dynamism stalls. A gerontocracy of the immortal could resist change indefinitely. Those who achieved power early would compound their advantages. New voices would struggle to be heard. Constitutional systems might need explicit term limits that outlive their original drafters, or mechanisms that periodically reset power distributions, or entirely new forms of governance suited to populations with radically different lifespans. These are not reasons to abandon longevity research. Death causes immense suffering. Aging robs people of their capabilities, their independence, their lives. The ethical case for fighting aging is strong, but the case is not uncomplicated. The downstream effects of success are vast and uncertain. We are contemplating a transformation of human existence more profound than any in history. We should proceed with eyes open. We should begin the policy conversations now. We should consider who will benefit and who will be left behind. We should think about what kind of society we want to become. The technologies are coming. The only question is whether we will be ready. And that brings us to the final question, the one everyone wants answered. When? When will we become immortal? The timelines, the predictions, the best guesses of those working to make it happen. This is where we conclude. When will we become immortal? This is the question that matters most. Not whether life extension is possible, not whether it is ethical, but when it will arrive. When will the technologies we have discussed actually become available? When could you, personally, benefit? The honest answer is that no one knows. Predicting technological timelines is notoriously difficult. Experts consistently underestimate how long hard problems take, and overestimate how quickly easy problems will be solved. The future does not arrive on schedule. But informed speculation is possible, and the researchers and futurists working on longevity have made their predictions. Let us examine them. Ray Kurzweil is the most optimistic mainstream voice. He has predicted that by the early 2030s, humanity will reach what he calls longevity, escape velocity. The point at which medical advances extend lifespan faster than time takes it away. If Kurzweil is right, someone who is 60 today might gain more than a year of life expectancy for each year that passes. Instead of approaching death, they would be pulling away from it. Each additional year of survival would bring new treatments that add more than a year of life. Kurzweil has a remarkable track record on technology predictions. He predicted the rise of the internet, the explosive growth of computing power, the emergence of smartphones, and the timeline for artificial intelligence advances with impressive accuracy. But he has also been wrong. His predictions about virtual reality and self-driving cars have not materialized on his originally stated timelines. And longevity is a different kind of challenge than computing. Biological systems are messier than digital ones. Still, his view is worth taking seriously. By 2029, he predicts AI will reach human-level intelligence. By 2035, he expects an age-reversing pill to be available. By 2045, the singularity, the merger of human and machine intelligence that makes virtually anything possible. If this timeline holds, most people alive today could potentially benefit. Someone in their 40s or 50s might reasonably expect to reach the escape velocity threshold if they maintain their health through conventional means. David Sinclair, the Harvard researcher whose work on epigenetic reprogramming has been foundational, offers a moderately optimistic view. He has predicted that age reversal therapies could be available within 10 to 15 years. His company, Life Biosciences, is actively developing treatments. Sinclair emphasizes that we already have interventions that extend healthy lifespan: exercise, caloric moderation, avoiding smoking, managing stress. These are not dramatic, but they buy time. The pharmacological options are expanding. Metformin and rapamycin analogues are available now, though not approved for longevity indications. Senolytics are in clinical trials. NAD precursors and other supplements show promise in animal studies. Sinclair's estimate suggests that someone who is 40 today might reach their 70s or 80s before meaningful age reversal becomes available. Those currently in their 60s or 70s face tighter windows. The race between individual aging and collective progress becomes very personal at those ages. Aubrey de Grey, the biogerontologist who founded the SENS Research Foundation, has long argued that the first person to live to 1000 is probably already alive. His framework focuses on periodically repairing the damage that accumulates with age, allowing indefinite extension. De Grey's estimates have been controversial. He has suggested that robust human rejuvenation could arrive by 2036, though he acknowledges this depends on adequate research funding. His organization pursues a specific program of damage repair therapies, targeting the fundamental causes of aging. Critics argue de Grey's timelines are unrealistic. The gap between laboratory, demonstrations and clinical therapies is large. Regulatory approval takes years. Manufacturing at scale presents challenges. But de Grey's fundamental insight may be correct even if his timeline is optimistic. Aging is damage. Damage can be repaired. The question is not whether but when and how. The more conservative view comes from mainstream gerontologists. Many academic researchers are skeptical of dramatic near-term breakthroughs. They point to the complexity of biological aging, the failures of past interventions, and the gap between animal models and human outcomes. A recent paper in Nature Aging argued that radical human life extension beyond current limits is implausible in the 21st century. The authors analysed historical data on maximum lifespan and found no evidence that the upper bound is shifting. Gains in average lifespan have come from reducing premature death, not from extending the maximum. This pessimistic view suggests that while we may continue to add years to average life expectancy, the fundamental limit around 120 years may be hard-coded into human biology. Breaking that barrier would require interventions more radical than anything currently in development. The truth probably lies somewhere between extreme optimism and fundamental pessimism. The pace of biological research has accelerated dramatically. Technologies that seemed distant a decade ago are now in clinical trials. Funding has increased by orders of magnitude. The smartest people in the world are working on the problem, but biology is hard. Translation from animals to humans frequently fails. Unexpected side effects emerge. Regulatory hurdles slow deployment. A reasonable middle estimate might be as follows. Within 5 to 10 years, we may see the first approved therapies targeting specific aspects of aging. Senolytics for particular conditions, gene therapies for age-related eye diseases, reprogramming approaches for localized tissue, regeneration. These will not be immortality pills. They will be treatments for specific problems, but they will demonstrate that aging can be intervened upon directly. Within 15 to 25 years, more comprehensive approaches may become available. Systemic senolytic treatments, partial reprogramming therapies for multiple organ systems, combination approaches that target several hallmarks of aging simultaneously. These could meaningfully extend healthy lifespan, perhaps 10 or 20 years, not immortality but substantial gains. Within 30 to 50 years, if current trends continue, we may approach something like longevity escape velocity, technologies that can reset biological age periodically, treatments that keep people at a biological age of 30 or 40 indefinitely. Beyond 50 years, prediction becomes nearly impossible. The technologies available a century from now are as unimaginable to us as smartphones were to people in 1890. What does this mean for you personally? If you're in your 20s or 30s, the odds are reasonably good that significant life extension technologies will become available during your expected natural lifespan. The key is reaching the early treatments, which then extend your runway to reach the next generation of treatments. If you're in your 40s or 50s, the race is tighter. You may benefit from early interventions, but you have less margin for error. Maximizing your health through conventional means becomes more critical. If you're in your 60s or 70s, the window is narrowest. The most transformative technologies may arrive too late, but early interventions might still add meaningful years, and cryonics remains an option for those willing to bet on more distant futures. What can you do now? The evidence-based interventions are well known if not glamorous. Regular exercise extends healthy lifespan more reliably than any pill. Cardiovascular exercise, strength training, maintaining mobility and function, diet matters, caloric moderation, nutrient density, avoiding processed foods. Time-restricted eating may offer benefits. The specific optimal diet remains debated, but the general principles are clear. Sleep is underrated. Chronic sleep deprivation accelerates aging. Quality sleep allows repair and restoration. Prioritizing rest is an investment in longevity. Social connection protects against decline. Loneliness accelerates aging at the cellular level. Maintaining relationships and community supports both mental and physical health. Avoiding obvious harms remains essential. Smoking remains the single largest modifiable risk factor for premature death. Excessive alcohol consumption causes damage. Obesity shortens lifespan and health span. These interventions are not exciting. They do not promise immortality, but they buy time. Time for the research to progress. Time for the treatments to arrive. Time to benefit from whatever the future brings. Some people are going further. The longevity optimization community experiments with supplements, drugs, and protocols beyond mainstream medicine. Metformin, rapamycin, NAD precursors, senolytic combinations. This is not medical advice. These interventions carry risks. Their long-term effects in healthy adults are unknown. Anyone pursuing such protocols should do so under medical supervision and with full awareness of the uncertainties, but the impulse is understandable. When the stakes are life and death, when the technologies seem tantalizingly close, waiting feels unbearable. The ultimate answer to when we will become immortal is that it depends on us, on the funding we direct to research, on the regulatory frameworks we create, on the distribution mechanisms we establish, on the ethical principles we uphold. The technologies are not inevitable. They require human effort, human resources, human choices. We could accelerate the timeline by prioritizing longevity research. We could delay it by underinvesting or overregulating. The generation alive today has a unique opportunity. We are close enough to see the possibilities clearly. We have the resources to pursue them seriously. We may be the first generation that can choose not to die, or we may fall short. We may reach the threshold of radical life extension just as our personal clocks run out. We may leave the gift of indefinite lifespan to those who come after. The outcome is not written. It depends on what happens in laboratories and boardrooms and legislatures over the coming decades. It depends on billions of individual decisions about health and resources and priorities. It depends in part on you. What will you do with this knowledge? How will you prepare for the possibilities ahead? How will you contribute to making them real? The dream of immortality is ancient, as old as human consciousness itself. Every generation has sought to defeat death. Every generation has failed, but we have tools our ancestors could not imagine. We understand biology at the molecular level. We can edit genes with precision. We can reprogram cells to younger states. We can eliminate the cellular debris that accumulates with age. We can interface brains with machines. We can preserve bodies for future revival. The dream that never dies may finally be within reach. The question is whether we will grasp it, and whether we are ready for what comes after. Thank you for joining me on this journey through the science of immortality. If you found value in this exploration, please like this video and subscribe to the channel. Your support makes these deep investigations possible. Tell me in the comments, if radical life extension becomes available, would you want it? How long would you choose to live? What would you do with centuries of existence? And which topic should we explore next? The simulation hypothesis, the future of artificial intelligence, the mysteries of consciousness, the possibilities are endless. Like the lifespan we may soon be able to choose. I will see you in the next investigation.